Welcome everyone to the 2021 USF Health Research Day. I'm Steve Liggett. I'm the Vice Dean for Research for the Morsani College of Medicine and Associate Vice President for Research for USF Health. And it is my great pleasure to have Research Day today. Things are a little different, as you might notice. It may look like I'm talking to you from our big auditorium where we normally have our Research Day begin, but that is not the case. I'm actually speaking to you from the new USF Health Heart Institute, and we are doing this event virtually. In the past, and I believe that we have still garnered the kind of enthusiasm that we want, we have set aside this day to celebrate research at USF Health. The take home when you leave Research Day has usually been that you've met some people, possibly even down the hall from you, who are doing research that has some collaborative potential with your own research. You've learned about what your neighbors are doing. You've interacted with multiple students and faculty at various levels across our 4 colleges. And we have presentations, judging, and awards. This year, obviously, we're going to have to do things a little bit differently. We are going to have and as always, we have a keynote speaker of renown, and we have that individual, Dr. Sessa, who I'll introduce in a moment, who will be coming to you virtually from Yale. As he presents his scientific lecture, we will be taking questions via Teams and working on triaging them so that we can have some questions at the end. His talk will last about 50 minutes, and we'll have between 10 and 15 minutes or more, if necessary, of discussion. And ultimately, when that is over, under normal circumstances, that's when all the activity begins. But we've actually had all the judging done. The posters have been viewed by the judges. We don't have the capacity to have each poster be viewed by all of the members who have joined us here on Teams. So we will just move directly to the award ceremony at that point. That would put us probably somewhere around 11 to 11:30. Where we'll be potentially finished. We'll just have to see. I would now like to go ahead and introduce our keynote speaker, Dr. William Sessa. I'll tell you a little bit about him. He obtained his bachelor's in pharmacy from the Philadelphia College of Pharmacy in 1984. He obtained a PhD in the Department of Pharmacology at New York Medical College in 1989. He did some short stints as a postdoctoral fellow and began his faculty career at the University of Virginia Health Sciences Center in 1992 and moved to Yale in 1993, where he has been part of the faculty of the Yale University School of Medicine ever since. And his area of study is really quite fascinating, and he is an excellent speaker and will be able to pull together years of his work to indicate new areas of research in cardiovascular diseases. To go into his career just a little bit, he is currently the Alfred Gilman Professor and Vice Chair in the Department of Pharmacology and Director of the Vascular Biology and Therapeutics Program at Yale University School of Medicine. His work defined the molecular aspects and the physiological implications of a molecule called endothelial nitric oxide synthase, or ENOS. And this is a master regulator of multiple biological processes, particularly in blood vessels. It controls how blood vessels grow, how they're permeable to salt and water, development of atherosclerosis, remodeling of the vasculature after injury. And so it's a very critical pathway. He, as a postdoctoral fellow, he actually cloned eNOS. And for those of you who were not in the cloning wars, you really missed out on something. Those were fun days. But what we, found out pretty quickly is that you can clone something, but you really need to figure out what it does, and at the, at a high level. And he has done that both in an integrative way and a very highly molecular manner. And so he's going to talk to us today about his work, and the specific title for his presentation is The Vascular Endothelium, Interfacing Lipids and Cardiovascular Risk. So Dr. Sessa, welcome to our research day. We're going to turn it over to you now.
Well, it's a pleasure to be with you today. I would have much preferred to have been in Tampa, but this is a new situation for all of us. So again, I'd like to thank Dr. Liggett and Sarah Yuan for inviting me to give this keynote presentation. So I'm going to give you a little overview of what we've been doing in the laboratory. Hang on. And give you an overview of the role of lipids in vascular function. And then some new vignettes of how to identify new targets that may impact lipid endothelial cell connections. Then I'm going to discuss the role of triglycerides and fatty acids on vascular homeostasis, and then just sort of a more philosophical approach to things that I've learned through medical research and things that medical research have taught me. Okay, so we know that out of all the leading causes of direct healthcare expenditures, cardiovascular disease accounts for about $196 billion of healthcare costs in the United States. And this was from 2015, so it's probably much more. And interestingly enough, there haven't been huge discoveries that have changed the course of events in the last 25 years, despite the fact that it's still the #1 killer in the Western world for all diseases. Now, when you look at cardiovascular disease, the major form of disease that causes death in people is coronary heart disease, which is really myocardial infarction secondary to atherosclerotic lesions in the coronary vasculature. And we know also that this is also occurring despite intense lipid lowering approaches, which we all know about. Blood pressure control, there's good antihypertensive medications out there. We know about stenting, we know about lifestyle modification, diet, et cetera, and still there's a huge impact of coronary heart disease in our population. Now, when you look at people that die from MI, abnormal lipids is the #1 risk factor for people that have died secondary to myocardial infarction. And there's very good data that has been generated through decades looking at the level of LDL cholesterol as a risk factor for coronary heart disease. And we know as the higher your levels of cholesterol go, the higher chance you have for having coronary heart disease. And a lot of the drugs which we use today, particularly statin-based drugs, have shifted the curve downward. So again, by lowering cholesterol, we tend to reduce relative risk. But in reality, even under optimal conditions of statin-based therapy or other therapies to lower LDL cholesterol, the relative risk reduction has only been about 30%. So it takes about 1 person out of 50 to be treated to have a protective effect against death. Okay, so let's talk a little bit about lipoproteins and lipids in the blood. I'm not going to bore you with this, but just to bring you up to the point where we are that's relevant to our work, we know that the liver is a primary source of endogenous cholesterol, and it produces cholesterol through, and it is carried through the particle VLDL, or very low density lipoprotein. The signature protein for VLDL is ApoB100, which decorates the outside of the lipoprotein, and you have cholesterol ester and triglyceride rich stores in the center of the lipoprotein. That lipoprotein then gets metabolized to form LDL particles. We know LDL is the major culprit of delivering cholesterol into peripheral vessels, such ones that will cause myocardial infarction in the heart as well as in the periphery. We know that cholesterol can be extracted from the peripheral tissues through the lipoprotein HDL, which brings cholesterol back to the liver. I want to focus a little on ApoB100 because this is really the signature protein that determines what's happening in the blood vessel. Now let's go back to the endothelium. So again, if you have lipids in blood, what is the first cell that is encountered? Well, it's the vascular endothelium. And we know a lot about the endothelium in the past 30 or 40 years. We know that under normal conditions, the endothelial cells in all of our blood vessels exert homeostatic functions. They control activation of platelets, it controls thrombosis, and also influences vascular tone. In the context of coagulation, we know that there are molecules on the endothelial cell surface that inactivate thrombin, which reduces the coagulation system. We know that there's tissue factor inhibitors, which blocks the activation of tissue factor, which is a major trigger for coagulation. We also know there are ATPases that will degrade circulating nucleotides, which are important for activating platelets. We also know that there's endothelial nitric oxide synthase, or NO, which can subserve an antiplatelet function. Also, there's vasoreactive prostaglandins like prostacyclin that will do the same thing. And we also know that there are these storage organelles called Weibel-Palade bodies that have von Willebrand factors and P-selectin. Under normal conditions, they are not exposed to the cell surface. In the context of vasomotion, we know that in most vessels, the nitric oxide pathway will cause vasorelaxation of smooth muscle, setting a biologic tone in the vessel. Whereas in the capillary circulation, there's tight junctions and adherence junctions that limit the flux of water from the capillary into tissue and allow for nutrient exchange. And finally, in post-capillary venules, where a lot of the inflammation takes place in response to damage and/or histamine release, we know that there's the trapping of leukocytes or neutrophils that will come into inflammatory sites. So the vascular endothelium really controls several aspects of flux of nutrients as well as cells into tissue. However, we do know that cardiovascular risk factors will cause endothelial dysfunction. So it's well established that diabetes, hypertension, hyperlipidemia, hypercholesterolemia, metabolic syndrome due to obesity, aging, as well as cigarette smoke, will all activate superoxide production from a variety of sources, which will then have an impact eventually on the ability of nitric oxide synthase to produce not just nitric oxide, but superoxide anions. So this idea that eNOS can be good under most conditions, then be uncoupled to generate superoxide has been established in the literature. And we know that endothelial dysfunction can promote heightened inflammation, enhanced platelet activation, and upregulation of coagulation pathways that can facilitate aspects of thrombosis. So where do we become interested in lipids in the endothelium? And it's really through a series of thought experiments that we did a while ago. When you think about what we know about the pathogenesis of atherosclerosis, and again, atherosclerosis is a vascular disease that's triggered by lipids in plasma. It's been agreed upon by many thought leaders in the field that it's the uptake of LDL across the endothelium and the trapping in the subendothelial space that provides sort of the kindling for the bonfire of the inflammatory response that takes place in the vessel wall. So it's believed that the subendothelial retention will allow for macrophages to phagocytize and oxidize the lipid, which will then activate an inflammatory cascade to bring cells into the vessel wall that will allow for the growing lesion. We know that there are cytokines that are elaborated by the macrophages and other cells that come in, such as TNF, IL-1 beta, that can also activate the endothelium to promote this sort of vicious cycle of LDL uptake. Now, when you have stable lesions, like most of us probably do over the age of 20, these are pretty much silent for a long time. However, as the plaque starts to grow, either due to necrosis of the core of the plaque or rupture of the shoulder regions of the plaque, you can then get the stimulation of a thrombus, which will then allow for a clot to occur, which will lead to an acute myocardial infarction. Conversely, if you do not have an unstable plaque, the plaque can still grow till it becomes a flow-limiting stenosis, which is about 80 to 90% occlusion, which will then result in chest pain. So we know that these two events are really the final determination of a growing plaque. So I'd like to go back to this initiating step. And this was really triggered by, so when I first came to Yale, I had to teach cell biology of blood vessels to medical students. And sort of the hero of cell biology at Yale was George Palade. So George Palade won the Nobel Prize for his utilization of the electron microscope to discover many of the organelles that we take for granted today. And he also worked a lot on blood vessels back in the 70s and 80s. And he wrote this monograph that I read probably about 15 years ago before my teaching. And it basically said, we know a lot about the chemistry of LDL and HDL, but we know much less about the way in which they reach the interstitial fluid in the cells that are living in it. And when I thought about the cells that were living in it, I was thinking primarily of the endothelial cells. So this really triggered a series of experiments really based on some of the original observations of Palade and his co-workers from a long time ago. So if you think about LDL particles in blood, and you think about the vasculature being the source of atherosclerosis, how does LDL get from the blood into tissue? So there are several proposed mechanisms, none of which have been proven unequivocally, is that you can have LDL particles that will bind to some receptor, maybe a scavenger receptor, which will then allow for what's called the transcytosis of LDL particles across the endothelium. And there's evidence that there's an organelle termed caveoli that can facilitate this sort of transcytotic event. The reality of this is that no one's ever shown a caveoli moving from apical to basolateral surface. Another possibility is that you can have a fluid phase transcytotic event that would allow for the movement of LDL particles not bound to any specific protein that would lead to accumulation of LDL. And the final thought was it's possible that LDL particles can traverse between junctions of endothelial cells of large vessels through a paracellular mechanism. But the evidence for this in animal models as well as in humans is not very good unless you have severe advanced atherosclerosis with disruption of the endothelium. So again, we started going into the literature and we wanted to understand this idea of how LDL gets across. And so the major fact of the matter is that it appears that the transcytosis of LDL occurs independent of the LDL receptor. The LDL receptor is the primary way to clear LDL from blood that's highly expressed in hepatocytes that allow for the clearance of LDL. As a matter of fact, that's how statin-based drugs work, by inducing the LDL receptor. But in the endothelium, the uptake for LDL transport is not through the LDL receptor. And there's several lines of evidence supporting that. One beautiful experiment that was published in the early 90s showed that if you make a mutation of the ApoB100 signature protein of LDL that will not allow for it to bind the LDL receptor, this mutant of ApoB100 is highly atherogenic in animal models, suggesting that you don't need to even have the LDL receptor in the endothelium for this to take place. One other interesting feature of this pathway is that the alternative pathway, the alternative to LDL receptor, is vesicular in nature. It's heparin sensitive, suggesting it's a charge effect, electrostatic, and occurs in the absence of lysosomal degradation of ApoB100, which is really a sine qua non of the LDL receptor pathway. When the LDL receptor takes up LDL, it degrades ApoB100 and recycles the receptor back to the cell surface. This alternative pathway does not do that. And this pathway is transcytotic, not paracellular. And when you look at the accumulation of LDL, it occurs at atherosclerotic lesions at sites of oscillatory flow dividers, which you know has dysregulated flow. And this occurs at bifurcation points of vessels. And so the question that we had probably about 10 years ago now was, how does LDL enter the endothelium when the LDL receptor is downregulated or not really important? And the reason we know it's downregulated is because as lipids go up in blood, through the negative feedback loop of the classic work of Brown and Goldstein, the LDL receptor is downregulated. And so the rationale to identify pathways is based on this observation. We know under normal cholesterol levels that there's binding of LDL to the LDL receptor. That gets taken in through a clathrin-dependent mechanism and delivers a cholesterol for normal cholesterol homeostasis for plasma membranes, for lipids, for things that happen within the endothelium. However, when you have elevated levels of LDL that occur during hyperlipidemic states, the LDL receptor is largely downregulated. This alternative pathway may lead to the accumulation of LDL cholesterol in the vessel wall. So this became the hypothesis. Okay, so to really get at this, we designed, or not we, I didn't do it, but several talented postdocs, John Chidlow, Jan Kraling, designed an assay where we can take fluorescent lipoproteins, fluorescent LDL, we can incubate them with endothelial cells under conditions by which the LDL receptor has been downregulated. Then we can knock down the entire genome using an siRNA library, and we can try to find genes that, when you knock them down, impact the uptake and accumulation of LDL in endothelial cells. We then looked at this data. We compared it to genome-wide studies and exomic sequencing data that were coming out of patient populations. And the idea was to try to understand, were there new molecules for mechanistic studies that we can perhaps develop a therapeutic strategy for, and even model in disease animals. So the idea was, can we find some new players that may impact LDL uptake in the blood vessels? So to make a long story short, I'm going to focus on one of those molecules today. And one of the hits that came out of our screen is the protein ALK1, or activin-like kinase. ALK1, depicted by this, I guess, black receptor over here, it's a type 1 serine threonine kinase that is a receptor for the ligands bone morphogenic protein 9 and bone morphogenic protein 10. This type 1 receptor will dimerize with the type 2 receptor that will lead to signaling from BMP9s to lead to the phosphorylation of SMADs that will then go to the nucleus to activate transcription of a variety of genes that maintain endothelial cell quiescence. And we know that either global knockout of ALK1 or endothelial-specific knockout of ALK1 is lethal due to both vascular and cardiac defects. So having physiologic levels of ALK1 to bind to BMP9 is critical for blood vessel function. We also know in humans that loss of function mutations of ALK1 can cause hereditary hemorrhagic telangiectasia of the type 2, which is depicted by arteriovenous malformations in the skin, in worst case scenarios in the brain, as well as forms of pulmonary hypertensions that are heritable. So again, the loss of function of the kinase domain will lead to these types of diseases. And the link between ALK1 and atherosclerosis really wasn't known, but there was one paper that suggested that ALK1 mRNA levels were higher in blood vessels of patients with coronary artery disease. So this became the beginning of an exploration for our group. So just to show you what ALK1 does to endothelial cells, this is a classic experiment from our original paper. If you take an endothelial cell line called EA926s or human umbilical vein endothelial cells, and you incubate them with di or fluorescent LDL, you get the nice uptake of LDL into the cells depicted by the red puncta that are in the cells. But if you knock down using siRNA ALK1 in either EA926s or in HUVEC, you can reduce the amount of uptake of LDL into the endothelium. And we knew this effect was specific because we could rescue this with overexpression of ALK1. And more importantly, if you take cells that lack the LDL receptor, you have the same phenomena. So this had nothing to do with the LDL receptor dependent pathway for uptake. Okay, so again, we knew that ALK1 was sort of a new receptor. The question was, well, if ALK1 is taking up LDL, where is the cholesterol going? And the cholesterol did not participate in normal cholesterol homeostasis, so we examined transcytosis. So to really look at transcytosis, and I think in the most quantitative way, we collaborated with Warren Lee, a scientist up in Toronto, and he developed a TIRF microscopy system, which is total internal reflectance microscopy, that can image LDL particles that can dock and fuse in the basolateral surface of the endothelium within 110 nanometers of the surface. So if you put labeled LDL on the outside of the vessel, you can follow its movement through the endothelium, so where the vesicle will then dock and fuse and release the contents to the basolateral side. And using this methodology, we were able to show that if you knock down ALK1, you reduce transcytosis to a significant extent. Moreover, if you overexpress ALK1, you can increase transcytosis, whereas overexpression of another family member of the ALK, ALK2, did not have such an impact. So this really showed that the effect of ALK1 was not just on uptake, but was actually on transcytosis of LDL particles. So the model which we have based on the literature and other people's work, that we know LDL will bind to the LDL receptor in endothelial cells, and this will provide cholesterol for normal cholesterol homeostasis, and the LDL receptor recycles. However, when cholesterol levels are very, very high, the LDL receptor becomes downregulated. This alternative pathway of transcytosis can occur. So LDL can then be taken up through the organelle caveolae, which ALK1 resides, as well as another receptor called SRB1, which will allow for the internalization and the endocytosis of ALK1, SRB1 positive vesicles that will then deliver cholesterol to the basolateral surface of the endothelium. So this is work that was done with Phil Shaw's work. Warren Lee have identified SRB1, we have identified ALK1, and work by our group and others have shown that caveolae are required for this internalization process. Okay, so to show some new data, so the question is, how do we dissect the importance of this pathway in vivo? And so I'm going to show you two approaches. These are both unpublished results, and it's very exciting for us because we think that there's a possibility that targeting ALK may be a new potential mechanism to reduce atherosclerosis. Okay, so the biggest problem we have with making a model to delete ALK was because if you delete ALK using conventional Cree reporter lines, those Crees will reduce ALK1 in arteries, veins, and capillaries. However, work by Ann Eichmann's group and Ralph Adams' group have shown that using a Cree called BMX Cree, which is largely expressed during development exclusively in the arterial endothelium, we could generate an arterial-specific ALK1 knockout animal that we bred to the LDL deficient animal, which has hyperlipidemia. So the idea was to take adult animals, use tamoxifen to inducibly excise BMX Cree in the arterial endothelium, and then feed the animals a high cholesterol diet for either 7 or 12 weeks, then examining LDL entry into the vessel wall and the extent of atherosclerosis. So the first experiment we did is we excised ALK1 in these ALK1 conditional animals, and then we injected the animals intravenously with fluorescent LDL, took out the blood vessel, and did en-face imaging by confocal microscopy to image the amount of puncta of di-labeled LDL in the arterial endothelium of the aorta in wild type as well as ALK1 deficient animals. And what you can see is that the loss of ALK1 in the arterial endothelium reduced di accumulation by about 60%. And again, there was no difference in lipid parameters in blood and lipoprotein profile, suggesting that the loss of ALK1 blocked LDL uptake into the vessel wall. More excitingly, if you feed the animals for 7 weeks on a Western diet, you can see this is now the thoracic aorta and the aortic arch of an animal that is hyperlipidemic. You can see the amount of neutral lipid depicted by oil red O staining in a lesser curvature, as well as in other vessels that come off the aortic arch. And this is highly atherosclerotic. And you can see in the animals that lack ALK1 in the endothelium, a marked reduction in atherosclerotic lesions. The same thing occurs after 12 weeks. This is after 12 weeks high-fat diet. This is an en-face image of the entire aorta of thoracic and abdominal aorta, as well as the branches. The red depicts the extent of atherosclerosis. You can see this is markedly reduced in mice that have reduced ALK1 in their arterial endothelium. So this really was great for us because, again, when we did the conditional knockout before with other Crees, such as the VE cadherin Cree, the animals would die after basically 2 weeks, even postnatally, which means that ALK1 is absolutely essential in veins as well as in capillaries. And we know that the HHT phenotype is largely due to ALK in veins and in capillaries, but not in arteries. Okay, so that shows genetically that ALK is important. Is there a way to neutralize ALK as potential therapeutic? And this is where it became very challenging, but it was also a very exciting approach. So the data that suggested this is possible was as follows. So in our original paper, we were able to immobilize native LDL onto a gold chip. And if you immobilize LDL, you can then have analytes that will flow through the chamber. You can get a binding event quantitatively using surface plasmon resonance. And when you do this, you can actually get these binding isotherms and you get on and off rates to show the level of binding. So we actually did this, and we used FC-ALK, FC-LDL receptor, and BMP9 to observe binding kinetics to the immobilized LDL particle. And when you do this experiment, you get the following binding isotherms. You can see that if you take LDL on the chip and you put FC-LDL receptor, you get a nice binding curve with a KD of about 7 nanomolar. If you do the same thing with FC-ALK, you can see you get a binding event, but with a lower affinity of about 200 nanomolar. But the interesting thing about these experiments is that if you have LDL receptor bound to LDL, it does not interfere with the binding of ALK1. And conversely, if ALK1 is bound to LDL, the LDL receptor will bind. And also, BMP9 did not compete for binding. So what this suggested to us is that there's non-competitive sites on the ApoB100 protein of LDL for both LDL receptor and ALK1. So with this in mind, we had the hypothesis, can we make an antibody that recognizes ApoB100 binding to ALK, but does not interfere with BMP9 signaling, which is essential for normal vascular function? So to do this, we collaborated with a company called Genovac. And Genovac was kind enough to develop a series of monoclonal antibodies for us. So they have these genetically modified rats and mice that have human heavy chain and light chain that have been genetically modified in the germline. So you then can take any antigen that you wanted to. So we took a human ALK1 ectodomain that was expressed on the surface of COS cells, and we immunized these animals. And then you can then take the spleens out and generate hybridomas like you would for making any type of monoclonal antibody. So the company generated 300 hybridomas for us, and they rescreened them against human, mouse, and monkey ALK1 and used human ALK5 as a counter screen. ALK5 is the canonical TGF-beta receptor. And they did this by flow cytometry, so they knew they were getting anything that would be on the cell surface. And from those 300 hybridomas, 89 candidates were delivered to us that were able to bind human, mouse, and monkey ALK1. So then in the lab, we had 2 postdocs that screened these candidates, and they were screened in endothelial cells using BMP9 as a ligand to activate SMAD1/5 in human endothelial cells. And so the assay that we used was as follows. We took UVEC, and then we incubated them with BMP9, and you can see activation of the SMAD phosphorylation. If you use a non-immune antibody, you can see that there's no effect. But if you use FC-ALK1 as a trap for the ligand BMP9, you can attenuate this signal. So what we did the exact same experiment, and we used all the different antibodies that were sent to us, and we looked for SMAD phosphorylation. And what you can see is that most of the antibodies were able to block BMP9 and do SMAD activation. Antibodies 11, 12, 15, 16, 17, and 18 all abrogated the ability of BMP9 to activate SMADs, whereas antibodies 13, 14, 19, 21, 22 did not. So out of these 89 antibodies, only 7 were confirmed not to block BMP9. So we then took these 7 antibodies and to see if they would block LDL uptake. And one particular antibody that we've been working with, antibody 2, was very efficient at blocking LDL uptake in vivo. So this is an experiment where we injected the antibody for 16 hours, let it circulate. Then the next day we came in and we injected a fluorescent LDL, let it circulate for 30 minutes. And then we imaged the amount of LDL after control antibody injection or anti-ALK antibody injection. And what you can see, there's a marked reduction in the amount of LDL that is taken up into the endothelium by the ALK1 neutralizing antibody. And the interesting part about this is a conformation selective ALK1 monoclonal antibody. So then we wanted to do an initiation study. So the idea was if LDL uptake and transcytosis initiates atherosclerosis, if we treat it early on with the antibody, could we impact the progression of atherosclerosis? So we took LDL receptor knockout animals, we gave them a Western diet for 2 weeks, and after 2 weeks, we then inject either PBS, non-immune IgG, or anti-ALK monoclonal antibody. We gave 2 injections a week for up to 4 weeks by giving them a Western diet. And then we isolated the aorta and looked at lipid parameters. And what you can see is that there was no effect of the monoclonal on weight, total cholesterol levels, or triglyceride levels in the animals. However, when you look at atherosclerosis, what we can see, again, this is only a 4-week high-fat feeding experiment. You get atherosclerosis by this en face image of oil red O in PBS control-injected animals, but much, much less in animals that were injected with the ALK1 monoclonal antibody. This is the quantitative data from these early studies. So this really supports our genetic evaluation of ALK1 being an important target. And again, neutralization of ALK1 during these early atheromas reduces the extent of atherosclerosis by about 30%. So we're now in the process of doing this for a long-term treatment study, and we're doing 16 weeks high-fat diet, and those results should be obtained by the end of March. Again, just to summarize this part of my work, or our work, the genome-wide screen identified ALK1 as a mediator of LDL transcytosis. And so to get to that point of even looking at ALK1 was really a ton of work, but I think we've come a long way since then. And I think the key experiment is that we know now the genetic loss of ALK1 arterial endothelial cells reduces atherogenesis, and this anti-ALK conformation-specific antibody that blocks LDL uptake but not BMP9 signaling looks promising as a strategy to reduce LDL in the vessel wall. And then from a conceptual perspective, I started reading a lot about this. We know very little about LDL transcytosis in general, about the molecules that take LDL from the apical to basolateral side, and also what regulates this process. So we've become very intrigued about this. And this is also relevant in the blood-brain barrier for other molecules, and in the capillary circulation, as well as in skeletal muscle for things like insulin. No one really knows how insulin's transcytosed across the endothelium, so we're starting to think more and more about these pathways. And then I guess from a conceptual 30,000 point of view, 30,000 foot point of view, can we reduce LDL transcytosis with anti-ALK antibody? And will this synergize with existing therapies for the treatment of coronary disease? And I'm really enamored by this idea because all the therapies that are out there clear LDL from the blood. This would really be the first example of treating the vessel wall directly to reduce the amount of LDL that gets across to initiate atherosclerosis. So we'll have to wait and see how that works out. But that's where we are now with this project. Okay, so the other area I'd like to just talk to you about really what was very exciting, I'd say, again, 5 years ago or 4 years ago, when, in the literature, and we've always talked about this to medical students and grad students, triglycerides are another lipid in blood, but it wasn't clear if they were considered atherogenic. And it wasn't until 2016 it became clear based on human genetics that indeed triglycerides are an independent risk factor for coronary disease. And the way this was discovered is as follows. We know that when triglycerides are in blood, they're carried in either chylomicrons or VLDL. And we know that the docking of these lipoproteins onto the enzyme lipoprotein lipase then allows for the lipolysis of the triglyceride to form free fatty acids and glycerol. And then the free fatty acids are then go into tissue and provide energy for skeletal muscle or for cardiac tissue. And we know that in the blood, there are certain negative regulatory molecules, such as ApoC3 and angiopoietin-like 4, which are negative regulators of lipoprotein lipase. And there's also positive regulators of lipoprotein lipase called ApoA5. And based on human genetics, there is evidence that loss of function mutations of LPL or loss of function of ApoA5 increase the risk, increase triglyceride levels in blood, and increase the risk of coronary artery disease independent of other risk factors. Moreover, LPL gain of function mutations or the loss of the negative regulators angiopoietin-like 4, ApoC3 reduce triglycerides in blood and decrease the risk of coronary artery disease. And so the question is, well, how do triglycerides link to coronary artery disease? Is it because the triglyceride remnants, such as chylomicron IDL remnants, also carry cholesterol? Or are there other ways, perhaps, that fatty acids and or lipolysis products can impact the risk of coronary disease? And again, so we really asked the question, and not, I say we, I mean Andrew Kuo, who was a graduate student in the lab, asked the question, is that we know very, very well in the microcirculation that the lipolysis of triglyceride-rich lipoproteins by LPL will release non-essential fatty acids such that will then be transported into the endothelium and transported into adipose tissue or taken up into skeletal muscle or cardiac tissue for energy. And there's no question that mutations that impact this pathway here have a large effect on plasma triglycerides, also skeletal muscle or cardiac function. But atherosclerosis is a large vessel disease. So the question we had to ourselves was, do large vessel endothelial cells process these triglyceride-rich lipoproteins? Or does this only occur in capillaries, since the docking protein GPI-HBP1 is uniquely expressed in the capillary endothelium? And the other question that we had is that, where do fatty acids go after you have lipolysis? When you have lipolysis that takes place in blood, we know that lipid is used for energy, but the entire circulation is exposed to the lipid, not just the skeletal muscle, which becomes a sink for the skeletal, for the lipid. Also, there was evidence in the literature, when you dig deeper, that you can find these lipid-dense bodies in hyperlipidemic animals as well as in humans. So this is an example of, you can see these lipid-rich cores that by electron microscopy that are in these blood vessels. If you look at this vessel here, you can see these opaque or these clear regions, which are lipid-containing droplets in the endothelium. You also see tons of them in smooth muscle, the underlying smooth muscle layer. And then finally, the evidence that you can get lipid droplet accumulation came out of a paper in 2008. This is a patient that has a loss of function mutation in the enzyme ATGL. ATGL is adipose triglyceride lipase that is not just expressed in adipose tissue, it's expressed in every cell of the body. And what you can see from this cross-section of a coronary vessel upon autopsy, you can see lipids that run through the entire intima, media, and the adventitial layer. And being an endothelial cell biologist, I sort of focused up here saying, wow, the endothelial cells are loaded with these lipid droplets. And so we became intrigued about, is it possible that lipids from blood get esterified into droplets in the endothelium, and that then becomes a source for lipid metabolism? I'm going to talk to you a little bit about lipid droplets. So there's very good evidence in all cell types that lipid droplets can be formed. And lipid droplets are formed using either chylomicron or VLDL-derived fatty acids as a source of substrate. These fatty acids are taken up through fatty acid transporters, such as CD36 or FATP proteins, where they then get into the cell and they get esterified with glycerol 3-phosphate through a series of enzymatic reactions, resulting in the formation of triglycerides that get esterified into lipid droplets. And the rate-limiting enzyme for the formation, or the final step in the pathway, is diacylglycerol acyl transferase, or DGAT. And there's two different flavors of DGAT that puts the final fatty acid onto the DAG molecule. There's DGAT1 and DGAT2, and this allows for the final formation of the lipid droplet. Now, the lipid droplet is a monolayer of phospholipid that buds from the bilayer of the endoplasmic reticulum, and it's thought that the lipid generation allows for the separation of the membrane to form a phospholipid monolayer with the triglyceride-rich lipid core. And the same thing can occur with cholesterol ester-driven lipid droplets, but by and large, we'll talk about fatty acid-derived droplets today. Now, when you make a droplet, there's also a series of enzymes that degrade the droplet. And as I told you before from that patient that was a homozygous mutant, the key rate-limiting enzyme in the breakdown of lipid droplets is ATGL. ATGL, along with this coactivator molecule CGI58, will provide the lipase activity to remove the first fatty acid from the triglyceride, followed by the subsequent hydrolysis by hormone-sensitive lipase, as well as MAG lipase, to release 3 fatty acids and glycerol. And we know that these fatty acid products are important for energy, phospholipid biosynthesis, as well as serving as lipid metabolites for PPARs and for other fatty acids that are important as ligands for nuclear receptors. So again, it's the sequential processing through this pathway. So we became interested, does this occur in the vasculature of large vessels? So the first experiment we did back in 2017 with Andrew Kuo, we took mice that we fasted overnight. We then gave them an olive oil gavage to increase the amount of triglycerides in blood, and we monitored triglyceride content as a function of time. And you can see as you give an olive oil gavage, after about 90 minutes, there's a peak of triglycerides in blood. These are largely chylomicron-rich triglycerides that then wanes over the course of the next 90 minutes. We then developed an on-phos imaging technique where you can take out the vessels at different time points, and you can image the vascular endothelium as well as the neutral lipid content using a Bo-Dipi dye that will then bind to the neutral lipid in the endothelium. And you can then monitor the formation of lipid droplets that you can see upon higher magnification are decorated in the endothelium of these large blood vessels. And quantify the number of lipid droplets per cross-sectional area. And you can see a peak in lipid droplets after about 180 minutes, which is the kinetic delay from what you see in the triglycerides. So what this suggests is that perhaps there's a lipid reservoir of droplets in the endothelium as a function of time. If you take endothelial cells in culture, you can do the same experiment. Instead of using olive oil, we use oleic acid, which is the major fatty acid, the monounsaturated fatty acid of olive oil. And you can see as you incubate endothelial cells with increasing concentrations of oleic acid, you can increase the amount of lipid droplets that are found in the endothelium. You can also do, this is a beautiful experiment where if you take endothelial cells and you load them overnight, if you remove the lipid for 24 or 48 hours, you get the loss of the lipid, and this tells you that there's active lipolysis. And if you use an inhibitor of ATGL, which is called ATGL-statin, you can see you can delay the lipolytic pathway depicted by the remaining green fluorescence that you see after the removal of oleic acid. You can also quantify triglycerides quantitatively. You can see if you load the cells with oleic acid, you get equal amounts of oleic acid in the droplets. And then as you remove, you see the time-dependent loss of triglyceride. And again, this is delayed by ATGL-statin. So it shows you that you can synthesize and degrade lipid droplets in large vessel endothelium. And this is sort of a nice preparation that we've developed in the laboratory. You can also do this ex vivo. So if you take a normal blood vessel from any animal, you can take a carotid artery, a femoral artery, or aorta. You can then incubate that vessel with oleic acid overnight. And then you can use confocal microscopy to image the endothelium depicted by PCAM versus the amount of lipid droplets determined by the Bo-Dipi dye. And what you can do is if you block DGAT1, which is the major synthetic enzyme for droplets, you completely eliminate the amount of droplets in the vessel. Conversely, if you use ATGL-statin, you can see that you can augment the amount of lipid droplets, and you can quantify this from these on-phos images. So again, it really shows that you can regulate the number of droplets in the endothelium. And so to summarize this paper from 2017, the concept was that if you have chylomicron or VLDL-derived triglycerides, when you have lipoprotein lipase, which will release the free fatty acid, those free fatty acids then could be taken up in the endothelium. And we know that free fatty acids such as oleic acid will protect from lipotoxicity driven by things like palmitic acid and other toxic lipids. We also know that these fatty acids that are derived from the droplets can also participate in fatty acid oxidations, because if you block LD degradation, you block the amount of energy or oxidation that takes place in mitochondria. And one thing that was really surprising is that LD-derived fatty acids can be released both abluminally or luminally. So it suggests that the fatty acids derived from the triglyceride can be released into tissue. Now, this is different from the direct vectorial transport of fatty acid across the endothelium, which happens within seconds. We're suggesting that perhaps lipid droplets can be a reservoir for lipid for subsequent usage later by the endothelium. Okay. So then this is all published data, but the question is, well, how do we test if these lipid droplets are dynamic in vivo, and what happens if we reduce the turnover in animals. So to do this, we generated a V-cadherin Cre-driven loss of ATGL in the endothelium, which we call the ATGL endothelial-specific knockout. This is work largely done by Anna Gamez, a postdoc who recently has left the lab. So first of all, this is, again, the thoracic and abdominal aorta. As you isolate them from animals, there's very few lipid droplets in normal animals. However, if you then give the animal oleic acid, you can start seeing the accumulation of lipid droplets in the endothelium of these vessels of control animals. However, if you take an ATGL knockout animal in the endothelium, you can see even as isolated, there's lipid droplets that accumulate. And if you give oleic acid, there's much, much more accumulation. So this suggests that the loss of ATGL in the endothelium will allow for the stabilization of lipid droplets in the endothelium. Again, this is very clear to see in cell culture. So if you isolate endothelial cells from the lungs of these animals, and then you put them into cell culture, you can see in the absence of any given fat, there's very few droplets in endothelial cells. And if you take ATGL knockout endothelial cells, even in the presence of normal serum, you find a large accumulation of droplets. However, if you give control cells lipid, you can see accumulation. And this is markedly augmented when you give ATGL knockout endothelial cells oleic acid. You can also measure triglyceride content directly. Again, the ATGL knockout is elevated at rest, but if you give wild-type cells oleic acid, it almost looks like an ATGL knockout. But if you give the ATGL knockout more oleic acid, again, you get larger accumulation of triglycerides. So again, the genetic loss of ATGL will allow for the accumulation of LDs. We then did RNA sequencing of these endothelial cells from wild-type ATGL knockout cells. We were able to we were quite surprised. And what we found was that there were many genes that are considered inflammatory genes that were markedly upregulated in ATGL knockout endothelial cells. And the major pathway that was activated was the CXCR4 pathway, leukocyte extravasation, which are the key pathways involved with inflammation that endothelial cells respond to. And if you look at some of the key markers, CCL2, which is MCP1, STF1 alpha is markedly elevated, tissue factor is up, suggesting procoagulant activity, COX2, or prostaglandin synthase 2, NF-kappa B is activated, VCAM1 is elevated. So again, the loss of ATGL in the absence of anything else that's happening in these cells in culture will promote inflammation or inflammatory gene expression. We also see an upregulation of genes involved in lipid metabolism, which I can talk to you about later. When we look at upstream regulators of these canonical pathways, it seems as if the TNF pathway, which again is mostly NF-kappa B dependent, which would also drive VCAM1, is one of the major pathways upregulated using RNA sequencing of these ATGL knockout cells. So then we wanted to ask the question, can we see evidence for inflammation in vivo? So these are experiments. If you take the cross-section of the aortic arch, we know that the lesser curvature of the aortic arch is more prone to atherogenesis. So this is cross-sections of the aortic arch, staining for CD31, which is endothelium, VCAM1, which is the inflammatory marker. And what you can see is that just the loss of ATGL in the endothelium increases the level of VCAM on the endothelial cell surface in the absence of hyperlipidemia, suggesting that the lipid droplets that are accumulating can promote inflammation. We also took endothelial cells from wild type or ATGL knockout cells. We stimulated them with LPS, looked at cell surface expression of VCAM by flow cytometry. And what you can see over here is that ATGL knockout endothelial cells have heightened expression of VCAM in the absence of LPS challenge. And this is markedly augmented when you give lipopolysaccharide to induce NF-kappa B. So again, it suggests that there's heightened inflammatory tone in the endothelium of ATGL knockout animals. We then looked at vessel function in these animals. So these are isolated aortic rings where we did the classic endothelium-dependent relaxation experiment. So we know if you take an intact vessel, if you give acetylcholine, Acetylcholine will cause a dose-dependent relaxation of the blood vessel due to activation of nitric oxide synthase. And this is, again, rightward shifted in the ATGL knockout vessels. If you look at phenylephrine, which is a vasoconstrictor, these curves are shifted to the left, suggesting a loss of sympathetic vasoconstricting, of buffering the sympathetic vasoconstrictive tone. Whereas if you look at nitroprusside, which directly relaxes the smooth muscle, there is no effect. So this suggests that the loss of ATGL is sufficient to trigger endothelial dysfunction. We then look for eNOS levels because eNOS is the major way to produce nitric oxide in these large vessels. This is a quantitative imaging of eNOS from wild type ATGL knockout endothelial cells, as well as from eNOS knockout vessels using on-phos imaging approaches. And you can see that in the ATGL knockout, that there's a marked reduction of perinuclear eNOS staining in these vessels as quantified over here. And we use eNOS knockouts as a control, which is a great control for the eNOS antibody. And you can see there's a marked reduction of eNOS protein levels in ATGL knockout endothelial cells. We've also bred the ATGL knockout to atherosclerotic animals, which are ApoE knockout animals. And what you can appreciate after this 14-week feeding, that there's a marked increase in the number of lesions that you find in these animals that lack ATGL in the endothelium. And this is very exciting because it suggests that the ATGL is a critical enzyme responsible not just in the capillaries for impacting fatty acid delivery for skeletal muscle or for cardiac tissue, but also in large vessel endothelium. So the concept that we think we're trying to develop here is that there appears to be fatty acid flux through the endothelium into lipid droplets that are happening very dynamically in the cardiovascular system, not just in the microcirculation, but also in large vessel endothelium. So we know as there's lipolysis and the formation of fatty acids that happen on a day-to-day basis, postprandially or during starving, that we know that those fatty acids can be incorporated into a lipid droplet, which then can be metabolized by ATGL into free fatty acids and glycerol. And this is normal for the normal maintenance of endothelial cell quiescence. However, if you knock out the gene for ATGL, there's accumulation of lipid droplets that drives a pro-inflammatory endothelial dysfunction that leads to reduced eNOS levels. So I think conceptually, it's actually very exciting because most people have only thought about fatty acid metabolism in the microcirculation, but we're making the argument that this happens very dynamically in all blood vessels in the body. So again, that's sort of the summary of my research, but I'd like to just give you some sort of, food for thought for those of you who are thinking about embarking on a career in research. And I really think, and you can talk to your professors, I think this is an unprecedented time for research right now. And there's no more or very little technological limitations. So I really think I would advise just use your imagination and think big. We can do single cell sequencing from any tissue, from humans and animal models. We can map all the genes, all the levels of expression during diseases. We can use CRISPR-Cas approaches in vivo and in vitro to really knock out any gene or modify any gene we wanted to, which is really incredible. So I think it's a great time to be doing research. And also, every medical breakthrough is achieved through basic and translational science. As we read these textbooks that are the fundamental textbooks in all the fields that we study, these are all based on basic research that we've done before. So the question that you have to ask yourself, do you want to be participating in the next generation of these new medical achievements? Which is really an exciting, sort of overwhelming thought as a scientist. And the other thing that I've learned through my training and by people that I've been mentored by, it's always important to surround yourself with colleagues who are intellectually curious, who challenge you. Anyone that tells you that you're doing well all the time is not challenging you. You want people that challenge your ideas to really push you to the next level. And I think this is so important. It's important to be done in a very constructive way where you have people that challenge you. The other thing that we think about sometimes is try to exercise both deductive and inductive reasoning with experimental strategies. And what I mean by that is that a lot of times we use hypotheses, but sometimes just random approaches using screening approaches is not a bad idea to give you new information to then test theories. And also, I think it's important to be a skeptic. I mean, don't trust everything that you read. It's always good to have doubts. And I think it's really the skepticism that really opens up new areas of research, because not everything is quite figured out yet, despite what textbooks tell you. And in the lab, one of my postdoctoral mentors, Sir John Vane, who was a Nobel Prize winner back in 1981, always used to say that good experimental strategies always yield results. It's just that we can't understand the results yet, because we don't really understand what the answers could be. And sometimes you don't like the answers, but biology never lies. It's just our lack of understanding of the answer that becomes frustrating sometimes. And I think the other thing to remember, especially during obviously COVID times, is try to enjoy the ride of science and the whole experimental process, and try to maintain some balance about what you do when you're in the laboratory. And my final slide is a thought slide here. I'm an avid golfer, and I'm a big Tiger Woods fan, and it's really horrible and timely that he had this horrible accident recently. But when I think about Tiger Woods' creativity and his greatness, these are two basically billboards for Accenture, who he worked for. And basically, you have information which is 40% of the situation, but it's really your interpretation that allows for innovation. And the other thing that's sort of thought-provoking is that when you're in the high grass, no matter how great you are, only 10% of what you did took you to this point. And it's really what you do for the next 90% that determines your ability to get out of this problem and to strive for excellence. So I think, with these thoughts in mind, I'd like to thank you for listening to my talk and entertain any questions that you may have. Thank you.
Thank you so much, Dr. Seth. That was a wonderful talk, and I think several points that you made towards the end are really important for all scientists to think about from time to time, especially our trainees, as we take them through the early and mid stages of their careers, to always be thinking in terms of some of the aspects that you were pointing out. We certainly don't want to get very comfortable with ourselves. Always be challenging ourselves and bringing ideas that may be technologically difficult, but are really necessary in order to take the field forward. We want to thank you so much for your wonderful talk and for participating with Research Day for us today. And we have a plaque for you. It's here. Are you going to put that on the screen? So this is your plaque, which will show up at some point. And it is our distinct pleasure to have had you here, and we will have you here when the epidemic, the pandemic is over, so that you can interact with us in the way that we're sort of more accustomed to. So thank you again very much. So there are a number of questions that have been sent in, and I encourage people to continue to send in questions as we work our way through. So do you have me off the screen now, Matt? Okay, so I have to move up close to the screen to read the questions. But I will start off with one from myself, and that is there a I don't hate to use the word magic. Is there a magic level of LDL that will, through which below you would not engage the ALK pathway?
I don't know the answer to that. That's a great question. I guess there's this idea that there's a constant flux of LDL across the endothelium. I think if the system needs cholesterol for membrane homeostasis, this alternative pathway would not be operational. So I think that's going to be the evolutionary conserve mechanism to take the cholesterol that you need for biomembranes, for example. So I don't know if you can go too low with this and lose that pathway. I guess it's testable. I think now with PCSK9 inhibitors, taking cholesterol to really, really low levels in patients, it's possible that this alternative pathway may not be as important. The one thing that's quite interesting is that we also have data that we can actually block the amount of ApoB that gets into the vessel wall, even under conditions where we have normal lipids. So I think there's always going to be a little bit of ApoB lipoproteins that get across, but I really don't know the answer. We don't know how far we can go with this.
Yeah, I think we've had we always think about once we find the pathway, well, let's just smash it down to nothing. And of course, the pathway is there for some positive reasons as well, and so we have to sort of think about that. A question from an anonymous person, so they must not want us to know what they do or do not know. Does LDL particle size matter in regards to the transcytosis or paracellular transit?
I'm sorry, my phone was ringing when you were answering that.
Oh, okay. Does LDL particle size matter in regards to this transcytosis or paracellular transit mechanism?
Absolutely. So at least there's a certain size, like LDL particles are about, I'd say, between 25 and 30 nanometers. And we know that LDL can undergo this transcytosis, as can VLDL remnants. In our hands, when we looked at HDL and oxidized LDL, they did not go through this pathway. But there's actually new data coming out from Ira Goldberg's group at NYU suggesting that the entire chylomicron remnant can be taken up by the endothelium. And this is not through ALK, because we've actually done some experiments with them. So there's other pathways that seem to take these entire particles up that are not the ALK pathway. But we know that SRB1 and ALK1 will take LDL particles up of the size of normal LDL, which is between 25 and 30 nanometers.
I'm going to scroll down. Sorry, someone put a PC in front of me, which is really not working for me. Is there any difference in the lipid droplet transport across a macrovascular versus a microvascular endothelium?
You know, I don't think so. I think what's happening is I think because when we look at, you know, the microvasculature in skeletal muscle from the animals that lack this ATGL, we do see lipid accumulation in the small vessel endothelium as well as the large vessel endothelium. So one point I want to make is that I do think when you have rapid lipolysis, there's definitely fatty acids that shoot across the endothelium, and those fatty acids are used for skeletal muscle function. There's no question that that happens independent of lipid droplet. The point I would like to make is that, yes, that is the case, but we know that there's also lipid that gets esterified into the LD, and that esterified droplet lipid could be a reservoir that you may need to either buffer the excess fat or to provide energy when you need it under conditions of a lack of lipolysis. So I think that there's two pathways that are involved here, but we see the same thing in both macro and microvascular endothelium.
All right, I think that that just adds an extra layer of complexity, of course, but that's what we love. Yeah. So this is a little bit of a correlate to the first question I asked, and this comes from Jerry Breslin. When you block LDL uptake, would that, first of all, does that lead to any elevation in plasma LDL levels? So is it a mechanism that is actually involved in clearance, which might be important? And then a follow-up would be, is there a potential way to accelerate the degradation of lipid droplets in the endothelial cells themselves?
Yeah, so great question, Jerry. So for the first part, when we've looked so far, at least in the EC knockout of ALK1, we haven't seen an effect on plasma levels, but we haven't of cholesterol or triglycerides, but we haven't done proper clearance studies yet. You know, the contribution of the LDL receptor in the endothelium to clearance has never really been looked at, to be honest with you. And so it's funny you asked that question, because we have conditional knockouts now for the LDL receptor in the endothelium, and we want to look at clearance from that perspective. I can tell you that when you think from a quantitative perspective, the amount of LDL that goes through the LDL receptor is at least 100 times more than can be taken up through the ALK protein as well as through SRB1. So, and from a quantitative perspective, this ALK pathway becomes operational as the LDL receptor pathway becomes diminished, which happens during hyperlipidemia in terms of net uptake. The interesting thing about the LDL receptor pathway is that it's not transcytotic LDL. It's only used for cholesterol metabolism. So there's very good evidence that the LDL receptor is not important for transcytosis, right? So you have this sort of low capacity pathway that's ALK1 and SRB1 dependent that does transcytosis. Then you have sort of the LDL receptor pathway, which does the bulk of the uptake, but does not contribute to transcytosis. In terms of accelerating ATGL, yeah, that's not a bad idea. I mean, you know, that's a good way to come up with an activator of that pathway. That actually might serve two functions. It may serve large functions in the endothelium of large vessels to reduce the inflammatory cascade, but also maybe it provides more fat for energy in skeletal and cardiac tissue. So again, it's a good idea. We haven't really thought about how to do that yet. What we're trying to do is block the pathway of LD formation. So we have animals that are now deficient in both DGAT1 and DGAT2, which is necessary to make a droplet. The complexity of that versus an activator is that the lipids got to go somewhere. So I wouldn't be surprised if we run into other problems if we can't make a droplet. So it's a good thought, Jerry.
Another question, which is fairly specific to one or two slides, is that one person felt that it looked like there was more accumulation of lipids in the ATGL deficient mice than the abdominal aorta compared to thoracic aorta. Is that the case, or?
Yeah, we always see that. We don't know why. I guess the implication would be is that there must be, maybe there's more ATGL expressed in the abdominal, so if you knock it out, you get more accumulation. We really don't know the answer to that, but we have noticed that, and it's not uniform throughout the entire vascular bed. Sometimes you see less or more depending on what vessel you look at, and I don't fully understand that. That's a very good observation. Yeah.
Another question, you didn't talk much about HDL. Is HDL involved with this pathway?
Yeah, so in terms of the ALK1 pathway, we have evidence that if you knock down ALK1, there's no effect on HDL uptake whatsoever. However, that stands in contrast to this SRB1 pathway, which is indeed an HDL receptor also. So it was actually interesting. There was a Nature paper in 2019 by Phil Shaw's group showing that if you knock out SRB1, you block LDL uptake. But there might be this competition between HDL and LDL for SRB1. We know HDL does not go through ALK1, but perhaps some of the cardioprotective effect of HDL is to block LDL binding to SRB1, right? So it's not unprecedented to imagine that possibility, but in terms of ALK, we have no evidence. It seems to be ApoB100 specific. That's why ALK1 works for VLDL as well as LDL.
So a potentially related question is the ALK1 pathway specific for LDL, or are there potentially other lipids or other proteins that are involved that utilize the pathway?
Yeah, we really don't know the answer to that. I mean, so we have in the lab I have a postdoc doing some mapping studies, and we can really map ApoB100 binding to ALK1, and we can resolve it to maybe about 100 amino acids that are critical for that interaction. And we think it's the signature protein ApoB that's determining the ALK binding event. It's not the lipid per se. It's actually the ApoB100 protein, which is sort of interesting. So, and we're trying to test the idea that chylomicrons can also bind to ALK1, because again, the first 48% of ApoB100 is ApoB48, which is the signature protein for chylomicrons. So we think it's actually a protein-protein interaction, not really a lipid-protein interaction.
Have you had it? I'm sorry to keep asking you questions, but you stirred the pot here, so we have to keep. So have you studied any sort of microvascular injury patterns in these mice?
Not yet. No. We just got the animals recently, and we haven't really done much with them besides what I showed you. But there is a collaborator and colleague at the University of Pennsylvania, Zolt Arani, who's a friend of mine. And Zolt and I have basically the same data for the ATGL knockouts, but he's done one other thing that we haven't done. The animals have impaired exercise tolerance, so it implies that perhaps this is important in the microcirculation in terms of the exercise response.
Excellent. Well, we want to thank you once again. I know that you're going to meet with some of the faculty later on in individual sessions, and so we'll see you at that time. Once more, I'd like to thank Dr. Sessa for coming and providing an excellent talk on our research day.
Thank you very much. It's a pleasure. I look forward to seeing you all in person.
So we're going to move on to the awards. This is so unusual feeling because we usually have this, as I said before, we have this period of beehive activity where people are looking at other people's posters, judges are integrated into the system in some way, and talking to the students, and rapidly having to ascertain the winners of our various categories, and we've already done that this year, so that it just makes it a little bit different. And I want to, as I go through each one of the each one of the awards and who has won the award, just everyone should know that you're going to get your certificate and your money if it's an award associated with a monetary component over the next few days. And the goal of our having this somewhat competitive is that we all know that science is competitive, and that we have a microcosm of individuals who we are collaborating with, but we're also somewhat in competition with. So with that spirit in mind, we do present a number of different awards. Some of them are very specific to the college of one of our 4 colleges within USF Health. Others have been partitioned out based off of basic science versus clinical or translational science, and some have been partitioned off as to the type of graduate student or postdoctoral fellow that is presenting the work. So we will begin with the medical student research awards. We divided this into years 1 and 2 and years 3 and 4, and these are our winners. All right. Now, the next series of awards are from all student types, so they were non-discriminatory in terms of what college or what level. These are the student research awards at the undergraduate level, master's level, and case study and chart review levels. Thank you, Heather. On to the basic sciences. We have an overall doctoral student research award, as well as the College of Public Health has one for their doctoral students and for a separate one for their master's students. And these are the awards. Thank you. We also have, as I mentioned, student clinical and translational sciences awards, and they are also broken down by an overall doctoral level students, and the College of Public Health has master's and doctoral students broken down. And these are the awards. Thank you, Heather. The College of Public Health has a Community Research Award, both at the doctoral and master's level, and these are the winners. The next series of awards is broken down by field, so you'll be able to see the different areas, and includes cancer biology, neuroscience, cardiovascular biology, etc. And these are all student types. So I put that up. Thank you. Got to leave it up long enough for people to look to see if they won, you know? All right. Now, from this point forward, the awards include the Lovelace Certificate, as well as a monetary award, which ranges between $500 and $1,000. And I will put the sponsor for each of these is included on the slide. The first one is the Morsani College of Medicine Outstanding Postdoctoral Scholar Research Award. Thank you. And then the next one is the Morsani College of Medicine Outstanding Resident Research Award. Our Office of Research at USF Health also sponsors an award which is called the USF Health Dean's Interdisciplinary Research Award that goes to Brian. Thank you. And Morsani College of Medicine Office of Research sponsors the Outstanding Innovations in Medicine Award. The Watson Clinic funds an Outstanding Fourth-Year Medical Student Poster Award, and this is our winner. And the USF Federal Credit Union has an award for the best poster in the field of immunology and microbiology. Tampa General Hospital, our flagship teaching affiliate and collaborator in research and clinical care, sponsors the Outstanding Clinical Fellow Award, and this is shown here. It's won by Benjamin Graham. So if I could have the last slide. I first want to thank all of our sponsors for providing the support necessary to put on this program. In addition, the staff that we have in the Office of Research does this and continues their regular day job. And it's not an easy task. It was not easy back when we did it in the big auditorium, and this time was a challenge as well. Our hope is that next year, at about the same time, we'll have our USF Health Research Day back in our usual location. I will say that we normally have in excess of 300 posters, which we did this year as well, and we look forward to seeing you next year. Thank you so much.